Welcome back to Anatomiqa. In the previous session, we completed discussing on the parts and neurovascular supply to the long bone. In this session, we will discuss on histology of bone. The learning outcomes of this session are: describe the microstructure of bone, list the bone cells and their functions, mention the microscopic classification of bones and their features, explain the features seen in the transverse section and longitudinal section of compact bone, mention the clinical significance. Bone is a specialized connective tissue. The cut section of the long bone shows the wall of the shaft is tubular and encloses a large marrow cavity. The wall of the tube is made of hard, dense material which appears smooth without cavities on naked eye examination. This part is called compact bone. The lining of marrow cavity and ends of long bones, diplo of skull bones, is filled with meshwork of tiny rods or plates of bone with numerous spaces in between. This type of bone is called spongy or cancellous bone. The entire bone on its outer surface, except the articular ends of long bone, is covered by a membrane called periosteum. The marrow cavity and the spaces of spongy bone are filled by highly vascular tissue called bone marrow. At the ends of the long bone, the marrow is red in color and contains numerous masses of blood-producing cells called hemopoietic tissue. In the shaft of adult long bone, yellow marrow is present, which is made predominantly by fat cells. In young child, the entire bone marrow is red, which with increasing age is replaced by yellow marrow. The wall of the marrow cavity is lined by vascular membrane called endosteum. Endosteum is one cell thick containing bone-forming cells, osteoblasts, bone-resorbing cells, osteoclasts, and resting cells, osteoprogenitor cells, capable of becoming active in response to appropriate stimuli. The resting form of osteoblast is flat and has squamous shape and is called bone lining cells. Let us recap what are the functions of bone. The bone matrix is mineralized. Bones become hard, dense and rigid. Because of mineral deposition in the matrix, bones can bear weight. It serves as a rigid skeleton of the body, provide sites of attachment for muscles and organs, provide protective cover for numerous soft organs. The brain is protected by the skull. The heart and lungs are surrounded by the thoracic rib cage, and the urinary and reproductive organs are located between the pelvic bones. In addition, the bones function in hemopoiesis, that is, blood cell formation, and serve as reservoirs for calcium, phosphate, and other minerals. Minerals can be either stored in the bones or released into body fluids when proper mineral homeostasis becomes essential. Almost all, that is 99% of the calcium in the body is stored in the bones. The body receives some of its daily calcium needs from this source. Calcium is essential for muscle contraction, blood coagulation, cell membrane permeability, transmission of nerve impulses, and other vital functions. Calcium release into the bloodstream from the bones or its deposition in the bones depends on the hormones released from either the parathyroid glands or the thyroid gland respectively. Bones are also dynamic structures. They are continually being renewed, remodeled, or both in response to mineral needs of the body, mechanical stresses put on the bone thinning during aging or disease, or healing during a fracture or break in the bone. As discussed earlier, the whole bone is covered outside by periosteum. It is highly vascular. Nutrition of underlying bone depends on the integrity of its blood vessels. Even after the development of bone has stopped, it has power to produce new bone when it is needed. Example, in the repair of fractures. When we take the section of the bone and observe under microscope, it consists of two layers of periosteum, outer fibrous layer which is composed of collagen fibers arranged parallel to the surface of the bone, inner cellular and vascular layer known as the osteogenic layer, It contains osteoblasts, which deposit layers of bones on the outer surface of the young bones. In adult long bone, the osteoprogenitor cells are present, and when need arises, as in case of fracture, they convert themselves into osteoblasts. Periosteum covers the whole bone except the articular surfaces of long bone in synovial joints, where it is reflected from articular margins to joint capsule of the joint. Let us see what are the functions of periosteum. It protects the bone, provides the attachment of muscles, ligaments and tendons. The fibers of tendon might continue obliquely at an angle into outer layer of bone as perforating fibers of Sharpie. It maintains the shape of bone and acts as limiting membrane. If periosteum is torn, osteogenic cells may extend into surrounding tissue, forming bony projections called exostosis. This is common in old age. It provides nutrition to the outer part of the compact bone by the periosteal vessels. It helps in subperiosteal deposits of bone formation, increasing the width of the bone during development. Coming to the composition of bone, it consists of extracellular or intercellular substance and the cells. Intercellular substance is also called as bone matrix. The intercellular substance of bone is composed of organic and inorganic components. Organic matrix or osteoid tissue makes up for 1/3 part of intercellular substance and consists of type 1 collagen fibers and ground substance. The type 1 collagen fibers are arranged in bundles and account for most of the organic contents. They provide tensile strength and flexibility to the bones. The ground or cement substance is homogeneous and comprises of glycosaminoglycans, proteoglycans and water. Proteoglycans, most important being chondroitin sulfate and structural glycoprotein, helps adhesion of bone cells with the collagen fibers. Inorganic matrix makes up for 2/3 of intercellular substance consisting of mineral salts. The most important constituents of bone salts are calcium, which accounts to 99% of inorganic matrix, sodium and magnesium accounting to 65%, phosphate accounting to 80%, and the rest being carbonate, chloride, fluoride and citrate. They provide enormous rigidity to the bone. If the inorganic calcium hydroxyapatite is removed by putting the bone in acid, it becomes flexible and can be tied as a knot. If organic tissue is removed by burning, the bone crumbles into small pieces. I repeat, the functions of organic and inorganic components. Inorganic calcium salts make it hard and rigid, which can afford resistance to compressive forces of weight bearing and impact forces of jumping. The organic connective tissue makes it tough and resilient or flexible, which can afford resistance to tensile forces. In strength, bone is comparable to iron and steel. Some elements like lead, plutonium and strontium may be taken up by bones and stored. The strontium may lead to malignant tumors of bones. Studies of strontium content of bones give information of radioactive fallout. 4 types of cells are recognized in bone tissue: osteoprogenitor cells, osteoblasts, osteocytes and osteoclasts. The osteoblasts, osteocytes and osteoclasts function like the Brahma, Vishnu and Maheshwara. Osteoblast forms the bone, osteocyte maintains the bone and osteoclast destroys the bone. Osteoprogenitor cells are pluripotent stem cells and derived from mesenchyme. In adult bone, progenitor cells lie beneath the periosteum and endosteum. The cells beneath the periosteum proliferate by mitosis and differentiate into osteoblasts, which increase the surface area of the bone by appositional method during development. The osteoprogenitor cells convert themselves into osteoblasts when needed, like in fracture repair. Limited osteoprogenitor cells proliferate by mitosis and differentiate into osteoblasts in more vascularized area with sufficient oxygen tension or into chondroblasts when oxygen tension is low. In the fetus, they are present numerously at sites where bone formation occurs. The osteoblasts are immature bone-forming cells. They are found lining the growing surfaces of bone. They are the precursor cells of the osteocytes and are derived or developed from the osteoprogenitor cells. Each osteoblast cell is cuboidal or columnar in shape, presents an abundant cytoplasm with large, round, eccentric euchromatic nucleus and numerous cytoplasmic processes. The cytoplasm is strongly basophilic due to ribosome granules attached to the endoplasmic reticulum. These help in the synthesis of bony matrix, both organic and inorganic. The osteoblasts are secretory cells and their cytoplasm, in addition to rough endoplasmic reticulum, contain Golgi apparatus, mitochondria and other organelles. Functions of osteoblasts. The main function of older or deeper osteoblast cells is to synthesize, secrete and deposit the organic components of bone matrix called osteoid around them. Osteoid is uncalcified, newly formed bone matrix that does not contain any minerals. However, shortly after its deposition, it undergoes rapid mineralization and becomes bone. These cells surrounded by matrix are converted into the osteocytes. The osteocytes are the main cells of the bone. They are derived from the osteoblasts and occupy spaces known as lacunae in the matrix. Only one osteocyte is found in each lacuna. There are flattened cells with centrally placed heterochromatic nucleus and numerous cytoplasmic processes which occupy the canaliculi and radiate from each lacuna and permit diffusion of nutritive material. The cytoplasm is eosinophilic due to negligible amount of secretory activity and small amount of endoplasmic reticulum. The cells remain alive in calcified matrix and secrete alkaline phosphatase to maintain calcification. When bone cells die, the matrix is decalcified. Average lifespan of an osteocyte is about 25 years. The osteocytes, like osteoblasts, do not divide by mitosis. Functions of osteocytes. They maintain the integrity of the lacunae and canaliculi and thus keep open the channels for diffusion of nutrition through bone. The osteocytes are responsible for the maintenance of the bone matrix. They play a role in removal of or deposition of matrix and calcium when required. Now coming to the third type of cell, that is osteoclasts. Osteoclasts are large cells that are found along the bone surfaces where resorption, remodeling and repair of bone take place. Because of this, the cells are called bone removing cells. Bone removal is essential for maintaining the proper shape of the growing bone. Osteoclasts are often located on the resorbed or enzymatically etched or eroded shallow depressions or pits in naked contact with the bone called resorption base or house ship's lacunae. Each osteoclast is a large cell varying from 20 to 100 micrometer in diameter. It possesses multiple nuclei with eosinophilic cytoplasm and is devoid of any processes. The cytoplasm is acidophilic, containing Golgi apparatus, numerous mitochondria, and numerous cytoplasmic vesicles or lysosomes filled with acid phosphatase and other hydrolytic enzymes. At sites of bone resorption, the surface of osteoclast shows many folds called ruffled membrane produced by numerous microvilli which abut on the bone surface. Electron micrograph shows that each osteoclast in Howship's lacuna possesses from within outwards ruffled border with microvilli projecting into the lacunae, clear zone called resorption lacuna which surrounds the ruffled border and seals off bone cell interface at the periphery by the attachments of actin filaments. The osteoclasts are derived from the fusion of the mononuclear phagocytic system or monocytes of the bone marrow, similar to the formation of giant cells by macrophages. After remodeling the bone by resorption, the syncytial cells of osteoclasts dissociate into mononuclear cells of the bone marrow. Functions of osteoclasts. The osteoclasts remove both the organic and inorganic components of the bony matrix. It appears that the primary function of the osteoclast is on the mineral, since these cells are not found in uncalcified bone. The osteoclasts secrete collagenase and other enzymes. To the subcellular sealed pocket, that is the clear space called resorption lacuna present between the ruffled border and bone cell interface, the cell pumps protons to provide a local acid environment. This promotes localized digestion of collagen and dissolves the calcium salt crystals. Before we go into actual structure of bone, let us see the classification of bone based on the microstructure. So based on microstructure, bone is classified into lamellar and non-lamellar bone. Lamellar bone includes compact and spongy bone from an adult skeleton. This is called mature or adult bone. On the other hand, the bone tissue which is initially deposited in the skeleton of the developing fetus is called immature bone or non-lamellar bone. It is also called oven bone. Compact cortical lamellar bone are seen in diaphysis of long bones, outer and inner table of skull bones. Cancellous, trabecular or spongy bone are present in epiphysis, bone marrow, short bones and diploe in skull bones. Non-lamellar bone is seen in developing fetus. Now let us see the details of microstructure of all the three one by one. Structure of immature bone or non-lamellar bone or woven bone. The picture is initial stages of developing bone. It does not display lamellar appearance and is called non-lamellar bone or bundle or woven bone because of interlacing arrangement of collagen fibers running randomly in different directions. It contains more cells per unit area than mature bone and are randomly arranged. The matrix has more ground substance than mature bone. It develops more rapidly than mature bone. Structure of spongy bone or trabecular bone. The left side picture shows the adult spongy bone. The right side is the diagrammatic representation of the section of the spongy bone. In spongy bone, the lamellae are arranged in the form of trabeculae or spicules. Trabeculae align along the position of stress. The trabeculae are irregular and form a meshwork or network in between which are numerous spaces called marrow spaces filled with bone marrow. The bony trabeculae consists of superimposed lamellae and usually do not form Haversian systems because they get nutrition from the blood vessels of the tissue around them. Therefore, the trabeculae are lamellated without Haversian system. The trabeculae are covered externally by vascular endosteum containing osteoblasts, osteoclasts and osteoprogenitor cells. It separates trabeculae from bone marrow. Peripherally, these trabeculae merge with a thin layer of compact bone which contains scattered osteons or Haversian systems. Coming to the microstructure of adult compact bone, when a small part of the shaft of the adult long bone is sectioned, it shows numerous units of bone structure called Haversian system or secondary osteons. These are cylindrical in shape, running longitudinally along the length of the bone with the long axis parallel to the long axis of the bone. They branch and anastomose with each other. Each Haversian system is about 150 micron meter in diameter and the central canal about 20 micron meter in width. Each Haversian system is demarcated from the neighboring systems by a refractile line called the cement line, which consists of modified matrix, strongly basophilic and is devoid of collagen fibers. About 21 million osteons are estimated to be present in the adult human skeleton. Each Haversian system consists of a central canal called Haversian canal surrounded by thin plates of layers of bone tissue or matrix. These thin plates of bony tissue are called lamellae. Successive lamellae differ in orientation of collagen fibers and in concentration of mineral salts. Accordingly, the lamellae are of two types, cementing and fibrillary. Cementing lamella are rich in minerals and less in collagen fibers. Fibrillary lamellae are rich in collagen fibers and less in minerals. Fiber arrangements in successive lamellae vary alternately. The collagen fibrils in a lamella are distributed parallel to each other but oriented in different direction when compared to adjacent lamella. Example, longitudinal, circumferential or spiral. With the advancement of age, longitudinal fibers increase in number and circumferential fibers decrease. The central canal of osteon contain reticular connective tissue, blood vessels and nerves. Anastomosis between central canals are frequently seen in the cross-section of the bone, but most commonly seen in the longitudinal section of the bone. Between the adjacent lamellae, numerous spaces called lacunae are present which communicate with one another and with the central canal by numerous radiating canaliculi. When a part of the adjacent lamellae and the lacunae is enlarged, it looks like this. As already discussed, compact bone is characterized by the arrangement of the bone matrix into thin layers called lamellae. Here in this picture, you can see two lamellae adjacent to each other, lamella 1 and 2. Between the adjacent lamellae is almond-shaped space called lacuna. In living bone, the lacuna lodges or contain the osteocyte. Because the mineralized bone matrix is much harder than cartilage, nutrients and metabolites cannot freely diffuse through it to the bone cells. Consequently, bones are highly vascular and exhibit a unique system of channels or canaliculi. These tiny canaliculi radiate from the lacunae in all the directions. Cytoplasmic processes from osteocytes extend into these canaliculi which radiate in all directions from each lacuna. The canaliculi contain extracellular fluid and the cytoplasmic extensions allow individual osteocytes to communicate with adjacent osteocytes and with materials present in the blood vessels that supply the bone matrix. In this manner, the canaliculi allow for a very efficient exchange mechanism. Nutrients are brought to the osteocytes. Gaseous exchange takes place between the blood and cells. Metabolic wastes are removed from the osteocytes. Some of the canaliculi open into aversion canals of the osteon and marrow cavities of the bone. Lamellae are arranged as following. Deep to periosteum is the outer circumferential lamellae. These lamellae are held together by the perforating fibers of Sharpie, which are derived from the periosteum and run obliquely, pinning the lamellae together. Lining the marrow cavity is the endosteum, which in turn is surrounded by inner circumferential lamellae. Between the outer and inner circumferential lamellae are the numerous cylindrically arranged Haversian systems. Each Haversian system or osteon consists of one Haversian canal and is surrounded by number of concentric lamellae. The small irregular areas or angular areas of bone between osteons are occupied by interstitial lamellae. Until now, we discussed the components and the general arrangement of the components in bone. Now we will study the specific arrangement of components of bone in two types of sections, transverse section and longitudinal section. In the transverse section of bone, the specific features observed are two layers of periosteum, outer fibrous and inner osteogenic layers, covering the whole surface of the bone. The marrow cavity is lined by endosteum. The bony matrix in the form of thin plates of lamellae are arranged in four types: concentric lamellae, interstitial lamellae, outer circumferential lamellae, inner circumferential lamellae. Okay, this has already been discussed. To recap, deep to periosteum is the outer circumferential lamellae. Lining of marrow cavity, the endosteum, is surrounded by inner circumferential lamellae. Between the outer and inner circumferential lamellae are the numerous cylindrically arranged Haversian systems. The Haversian systems or osteons are seen in cross sections with Haversian canals, which are longitudinal channels lodging the reticular connective tissue, blood vessels and nerves. Each Haversian system or osteon consists of number of concentric lamellae surrounding the Haversian canal. Small irregular areas or angular areas of bone between the Haversian systems or osteons are occupied by interstitial lamellae. This is the photograph of the transverse section of compact bone as seen under the scanner of the microscope. The structure may not be exactly observed as discussed in the previous slides. Once you start identifying and master the sections, you will know the structure properly. So in this section, what you can see is many osteons or Haversian systems can be identified with the Haversian canals in the center. Concentric lamellae can be observed surrounding the Haversian canals. In between the Haversian systems, interstitial lamellae is seen. Sometimes, very rarely in the transverse section, the connection between the two Haversian canals can be observed if the plane of section passes exactly at the level of connection. This connecting channel is the Oakman's canal. This canal is commonly seen in longitudinal section, but sometimes like this can be also seen in transverse section. This is the photograph of the transverse section of compact bone as seen under the low power of the microscope. The previous section was under the scanner. All the features as seen under the scanner are observed even in the low power view. Many osteons or Haversian systems can be identified with the Haversian canals in the center. Concentric lamellae can be observed surrounding the Haversian canals. In between the Haversian systems, interstitial lamellae is seen. Sometimes, very rarely in the transverse section, connection between two Haversian canals can be observed if the plane of section passes exactly at the level of connection, which is the Oakman's canal. This is the photograph of the transverse section of compact bone as seen under the high power of the microscope. Enlarged view of a part of one osteon with the details are observed in this section. One Haversian canal can be observed in the right lower corner of the picture. This Haversian canal is surrounded by thin plates of concentric bony lamellae. In between the bony lamellae or the spaces, the lacunae, which in the living lodges the osteocytes. From the lacunae, thin canals called canaliculi radiates from all sides. Okay, these are few other photographic sections observed under the microscope for you to identify the features. The next 3 slides shows different transverse sections of the compact bone. Try identifying the Haversian system, Haversian canal, concentric lamellae, interstitial lamellae and lacunae. We have seen the photographs of the unstained sections of the bone until now. They are called ground sections. Since the bone is hard, very small pieces of bone is sectioned and ground on a stone to a very thin slice so that it can be observed under the microscope. Usually only unstained section is kept for identification. This is one of the way you can identify the bone for spotters. This section, what is observed is the decalcified section of bone. A small piece of bone is cut and immersed in the acid for several days until the calcium is digested completely. Then, this piece of bone is processed as usual like any other tissue or organ in the body, sectioned and stained with H&D or with the special stains. This is another photograph of the decalcified bone section stained with H&D. Until now, we discussed the features of transverse section. Now let us see what are the features observed in longitudinal section of compact bone. In the longitudinal section of bone, the specific features observed are bone is covered by periosteum and is lined by endosteum. 2 layers of periosteum, outer fibrous and inner osteogenic layers covering the whole surface of the bone. Marrow cavity is lined by endosteum. Deep to periosteum is the outer circumferential lamellae. Lining of the marrow cavity, the endosteum, is surrounded by the inner circumferential lamellae. Haversian systems or osteons are seen in longitudinal sections with Haversian canals running longitudinally, lodging the reticular connective tissue, blood vessels and nerves. The thin plates of bony lamellae are arranged parallel to longitudinal sections of Haversian canal. Lacunae between the adjacent lamellae are occupied by osteocytes in the living. Adjacent Haversian canals are connected with each other through transverse channel called Oakman's canal. Oakman's canals, apart from connecting the Haversian canals, also communicate with medullary cavity and with the outer surface of bone. Both Haversian and Oakman's canals contain blood vessels and nerves which permeate the bones from the periosteum or endosteum. This is the photograph of the longitudinal section of compact bone as seen under the scanner of the microscope. In this picture, longitudinally cut sections of Haversian canals can be observed. Adjacent Haversian canals connected by transverse channels, Oakman's canal, can be identified. Lamellae arranged parallel to Haversian canals also can be seen. So here you will not identify the different types of lamellae as seen in transverse section. It is only the parallel arrangement of the lamellae which can be observed. This is the photograph of the longitudinal section of compact bone as seen under the high power of the microscope. Enlarged view of a part of two adjacent Haversian canals connected by transverse channel Oakman's canal can be viewed. Thin bony plates, lamellae, arranged parallel to Haversian canals is identified. In between the lamellae, the spaces called lacunae, which lodges the osteocytes in living, are visible. In this session, till now, we have discussed the introduction, functions, components, general arrangement of the components, specific arrangement of the components in transverse and longitudinal sections of the bone were discussed. In the next session, I will be discussing on bone fracture and repair.